Hi, I'm JD Tamucci. I'm a TA for this Biology 1107. I'm going to be talking about what to study for Practical 1. So, first thing you should know is that for Biology 1107, on the practical, there are 26 questions, including one bonus question. So, since one of them is a bonus question, say 25, but then there's going to be some questions that don't relate to any specific lab, like a practical question. And since it won't relate to a specific lab, we'll subtract some of those. So figure like 20-ish divided by 7 labs. You're talking about 2 to 3 questions per lab. So that's what you should expect. Now, going into studying, you should then think about, like, all right, here's my lab manual. Here is, all the information from that lab. If I could only ask 2 to 3 questions about this lab, what would I ask? That's how you should approach it, because that's how we had to approach it when we made the practical. So come up with variants of what you think would be on there. But to help guide your studying, I made this. Okay, we're going to have come to office hours to better understand this, but I'll go over some things briefly. I'm not going to go over specifics of which things you should study, but this should guide your studying as to the general categories of what we make questions out of. So, reagents. Anytime we use a reagent like BSA or DCPIP or isopropanol or whatever, SDS, we're using it for a reason. You should know why we're using it, how it plays into that lab, and yeah, that you should be able to explain it, reproduce it on an assessment. Math. Every math problem we do is fair game and is probably going to show up on an assessment. If you haven't noticed that already, on all the quizzes, if you did a math question in a lab, it ended up on the quiz. That's just how it works. That's how we test your arithmetic for the most part. It's usually just algebra, okay? So all the math questions from concentration to, actually no, that's all we do in this half of the semester. Concentration for the most part. The third lab, because the third lab people seem to forget about. Remember, we looked at electron micrographs of organelles. We had to know what their functions were. We used a microscope to look at like protists and bacteria and human cheek cells. We also used the what did we do? We did tonicity. We looked at all of the dialysis tubing, looking at how sucrose couldn't pass the dialysis membrane, but water could, and how things would, cells would react differently to different solutions that they're in. So that's another thing that is important that a lot of people forget about, but you can expect to see on the practical. Data in general. So this is something that doesn't always relate to a specific lab, but could relate to many different ones. So you need to be able to present data. If I were to give you something like, hey, I need you to make a graph out of this data from a table, I would expect it to look good. How to interpret data. If I gave you a graph, we went over all kinds of different graphs, like standard curves, the absorption spectrum, what else? All of the graphs from the fermentation lab, a DNA standard curve, when we used a semi-log graph, which was kind of new for us. You need to know how to make those, how to interpret the data, how to look at a graph and say, all right, this is the important thing that I would need to talk about. That's one thing we expect. And then understand why we use that graph. Why do we use an absorption spectrum? What about a standard curve? Why did we use the fermentation temperature graph to isolate what the optimum temperature is? And then that kind of links into lab report skills. So you should know how to write a proper hypothesis, what goes into an intro. Remember, you get to put your hypothesis at the end of the intro, and then your intro kind of functions as everything you would need to understand your hypothesis. So I get general background, and then it sets up your hypothesis. And then the discussion. Remember, first sentence, you're talking about, did your hypothesis get supported? And then you're going to support that data. And then talk about other trends, comment on sources of error, those type of types of things. And then the only thing I'll say about figures and tables is remember where the legends go. A figure legend goes below the figure. A table legend goes above the table. So I don't know if I mixed table legend above, figure legend below. Hopefully that makes sense. All right, and then practical questions. There we go. Okay. These things, I'm not the greatest artist, but there are labels. So, pipette, that is a micropipette, and it could also be a serological pipette. We'll say it's both. You should know how to do that. Microscope questions. Parts of a microscope, how to use a microscope, all those things. Totally fair game. That's a scale. Remember we use a scale to measure things, how to tare it. That's also fair game. What else? And then the spectrophotometer I couldn't draw, but that's something we used a lot throughout the semester. You should also be able to use that. Why do we use it? Why do we use these things? What are they for? What can they allow us to do? Colorimetric shifts, get better resolution of something, be more accurate when measuring something. Those are the reasons for the most part. And then if all else fails, look at the lab objectives. The way that we made this the lab objectives can always direct your studying. They're available on Husky CT at the beginning of every week section, or lab week section, and from them, it kind of directs you to understand what we did, why we did it, and how we did it. Those are the main things that if you're able to succinctly explain those things, then you're probably going to do pretty well on the practical. But yeah, so that's everything. Good luck studying, and if you have questions, come to office hours.